Now we will talk about regulation of cell cycle. There are four checkpoints present in the cell cycle: G1S checkpoint, intra-S checkpoint, and G2 or M checkpoint, right? And the last one is spindle assembly checkpoint, which is present between metaphase and anaphase. As you can see here, this is the G1S checkpoint. This is the intra-S checkpoint, which is present in the S stage only, S phase only. This is the G2 or M checkpoint. This is the spindle assembly checkpoint, which is present between metaphase and anaphase. Now we will talk about these checkpoints. Now, suppose if the cell is ready, right, for a round of cell cycle, then G1S checkpoint is the first one. Now, first I will talk about even before the G1 or S checkpoint, right? There is a stage called G naught stage, or also known as regulatory stage. So what happens is, even before the starting of the cell cycle, this G1 or S checkpoint is very important, because if it recognizes any problem, it receives any problem or signal that there is some problem in the cell, then it will keep the cell in a G naught stage, which is also known as regulatory stage. In this stage, the cell will not divide, and some cells will remain in the G naught stage for the rest of their life, just like nerve cells of our body. Okay, now we will talk about G1 checkpoint. G1 checkpoint involve PRB or E2F genes. It involves PRB, retinoblastoma pocket protein, or retinoblastoma sensitivity protein, whatever it is. And second is E2F, E2F gene. E2F gene, it is a group of genes which code for transcription factors. It regulates transcription factors. We'll talk more about E2F further now. And there is also another set of or complex of proteins or factors, which is CDK protein, CDK4, CDK factor 4 or 6, along with cyclin D. These are the proteins or factors which are responsible for G1 or S checkpoint. Right. Now we will talk about intra-S checkpoint, a little about intra-S checkpoint. Now, what G1 or S checkpoint did was it checked whether the proteins are ready for the replication or not. Now, replication is ongoing in the S phase, right? Now, it will check whether the DNA replication is going properly or not. G1 or S checkpoint check whether it will be all the proteins which are required for the DNA replication are ready or not. Then S checkpoint, intra-S checkpoint check whether that process, that is the replication, is going properly or not. Now, what are the factors involved in the intra-S checkpoint? Intra-S checkpoint, there are two sensory mechanism, ATM or ATR sensory mechanism. These are sensory mechanisms which are play a key role in the DNA death response, DDR. You can say they play a key part in the DDR, right? And there is another one, yes, along with CHK1 and CHK2. They also play a vital role. They regulate the cell cycle. Just remember this only right now. Now G2M checkpoint. Now after DNA replication is done, right? DNA replication is done, S phase is gone. Now comes the G2N or M checkpoint. What it does, check test if there is any DNA damage or not. DNA is replicated, then that formed replicated DNA has any damage or not, right? If there is any DNA damage, then CHK2 will be activated, or it can also activate P53. P53. What is P53? P53 is the guardian of the cell cycle, right? P53 is the guardian of the cell cycle. We'll talk more about in depth about it afterwards. In this video only, obviously. Now spindle assembly. Spindle assembly checkpoint. Spindle assembly checkpoints check whether the mitotic spindle from opposite pole are properly connected to the kinetochore or not. That is, the tension coming from both the poles are equal or not. And if one side is lacking the tension, then that is a negative signal, right? That is a bad signal, right? That means that the checkpoint will stop that signal. Right? It means that checkpoint will stop the cell cycle at this point. It gets a negative signal, right? As we know that kinetochore, the structure between the center point of the position of the chromosome, right? Chromatid, the sister chromatid, which is attached to the opposite poles with the help of mitotic spindle or microtubules. Now, what is it involved? Spindle assembly checkpoint involves securin and separase. Securin and separase, we'll talk more about it afterwards. Now, so we will talk a little more about P53. P53 in turn recruits P21. P21 inhibits cyclin. Thus, it is a cyclin inhibitor. Then, after inhibiting cyclin, it inhibits the cyclins are inhibited, which inhibits the which inhibits the cell cycle, or halts the cell cycle, or stop the cell cycle. Halts. It will not progress further. This is the role of P53. Or what P53 does is P53 try to solve this problem or fix the problem if there is some DNA damage, right? Single stranded DNA damage or double stranded damage. P53 will be activated, and they try to solve the problem with the DNA repair mechanism. There are different DNA repair mechanisms. Nucleotide excision repair, different base excision repair. There are different repair mechanisms, right? And if they fail to do so, they will finally trigger another pathway, death pathway, right? Death pathway, which is also known as apoptosis or programmed cell death, that will kill the cell deliberately. Now we will talk about vital functions of different checkpoints. We'll mainly talk about two checkpoints: G1S checkpoint, and second one is spindle assembly checkpoint. In G1S checkpoint, E2F is very important. Right? E2F, a group of genes, encodes the transcription factor, which encodes the transcription factors. Different E2F genes are there, like E2F1, E2F2, E2F3A, E2F3B, which are further classified as activators and suppressors. Now, E2F is very important. What it does is it allows the synthesis of enzymes, right? Transcription factors, which is encoded by ETF. It allows the synthesis of enzymes which are required for DNA replication. Now, E2F is very necessary to move from G1 to S phase. Otherwise, it cannot be done. Right? By default, this E2F protein, what happens is that it is blocked by a protein known as PRP, retinoblastoma sensitivity protein, or retinoblastoma pocket protein. It looks somehow like this. Suppose this is the E2F. What happens is it is blocked by this PRP protein, retinoblastoma pocket protein. Now, we can say that PRP masks the active site of this E2F, and this blocks it. Now, what happens is complex comes CDK. Let me change. CDK 4 or 6 along with cyclin D. It comes here. What it does is this complex phosphorylates. It phosphorylates what? PRB. PRB protein. It phosphorylates PRB protein, which makes it inactive. It becomes inactive, and then they will tag the polyubiquitin and they will degrade the PRB. As a result, the E2F becomes free and active and can do all the work, that is production of the replication enzymes. That is, the cell is now ready to progress from G1 to S phase. Right? What will happen? It inactivates it inactivates this, and what will happen is this becomes free, E2F becomes free and becomes active, becomes free and active, right? Which will result in production of replication enzymes, right? So we will now talk about spindle assembly checkpoint. Now spindle assembly checkpoint, here you can easily see how it happened, right? After it becomes active, it produces a replication enzymes which are very necessary to move from the G1 to S phase. So what does spindle assembly checkpoint do? Spindle assembly checkpoint regulates whether the segregation of sister chromatids is done or not, whether they are separated or not, right? And the signal is on when the tension coming from the opposite poles are equal to the kinetochore. We talk about it a little bit. Here is the tension to the kinetochore coming from the opposite poles are equal, then the signal will be on, that there is no problem and the cell cycle can continue. But if the tension is lacking from either of the sides, then that is an alarming signal, right? The checkpoint will stop the movement of the cell cycle, and once it puts a ubiquitin tag on the securin, sooner or later securin will be destroyed, right? So in spindle assembly checkpoint regulates whether the segregation of or separation of cystic chromatids will be done or not, and the signal is on when the tension coming from the opposite poles are equal to the kinetochore, and if the tension is lacking from either of the sides, right? If there is equal tension from the kinetochore, from coming to the kinetochore from the opposite poles are equal, then the signal is on, right? That means that cell cycle can progress without any complications, without any problems. But if the signal is off, what does it mean? That it means that the tension that is coming from opposite poles is not equal, right? And it is an alarming signal. The checkpoint will stop the cell cycle from progressing further. The checkpoint will stop the cell cycle from progressing further from metaphase to anaphase, and the cell cycle will halt. Now, as we know that cohesin, there is an enzyme or a factor that holds the cystic chromatids together, right? It is known as cohesin. Now, this cohesin can be destroyed or degraded by an enzyme known as separase, and as a result of which, the cystic chromatids will be separated. Now, the separase is present in the cell, but it is not active, right? It is blocked by securin. Like this. Suppose this is separase. What happens? Is it is present in the cell, but it is present in the inactive. It is not active. It is an inactive state, which is why, because it is blocked by securin. Somehow like this. This is securin. It blocks the separase. So what will happen? Does the securin prevent the separase from destroying the cohesin? Right. This is the cohesin. Suppose this was the cohesin. Now what will happen? That securin prevents the separase from destroying the cohesin. It will not be able to go there. It will be blocked by securin, due to which cohesin is intact and the sister chromatids are also intact. Right? As we know that metaphase plate is organized and the chromatids are present on the metaphase plate, we all know that thing, right? Now what will happen after this? Now what will happen after this? Suppose there is some factor which makes this also fail, this also fails, right? Suppose this process also fails, right? Now if a proper connection is made, then a complex of protein known as APC gets activated, which in turn polyubiquitinate the securin. Suppose what will happen, a complex of proteins APC comes, right? Suppose APC comes, it polyubiquitinate this securin, which in turn will degrade the securin, due to which what will happen, this separase will be free. This separase become free, due to which it can now easily attack cohesin. That cohesin now is in danger. Is when this separase gets free and active, it will degrade the cohesin. It will degrade the cohesin, right? Due to which sister chromatids will not be intact, right? And if this also fails, then what will happen? Cell cycle gets arrested. Ultimately, if there is any problem in this any of the checkpoints, the cell cycle will get inhibits and everything will fail, right? Right? Cell cycle will halt. Cell cycle, which is known as cell cycle arrest, which will ultimately lead to the death of the normal cell, or it may convert normal cell into cancerous cell. Right here you can see. If this also fails, then the cell cycle gets arrested, and all the checkpoints are necessary for. And if they fail, the normal cell will convert into cancerous cells. Thank you very much.